So, as I said before, there's four themes, and our first theme for today is eat, sleep, rave, repeat. Because this is, after all, Tomorrowland. And here to host the first part is Dr. Nadine Bongaerts.
Just say Nadine now, it's okay.
Okay, Nadine. A synthetic biologist who believes biology will drive innovation in the coming decades. Why do you believe that is the case, Nadine?
Well, that's what I'm going to tell the audience in my talk. So
okay, that's my cue.
Yes.
Bye.
Bye-bye.
Have fun.
Okay, Hello Love Tomorrow. Wow, I've never been on a stage like this. This is incredible. What an opportunity to celebrate sustainability. It's a big, big honor for me to stand here in front of you and to present and host this very first session of the conference, in which we will show you that sustainability is scalable and that it doesn't impact the lives of just a few, but of many. Now, I don't know if the organizers picked this date on purpose, and Nina already said it. Today is Earth Overshoot Day. It means that we officially used up all the resources that the planet regenerates in a single year. To meet the demands of our current lifestyle, we don't need one, but actually 1.75 planets. Now, unless there's someone who can build us a second planet, this is pretty scary. And yet, this is also the time to be hopeful and to take positive action on the many opportunities that are out there to turn things around. Because new innovations and smarter and better ways of producing food, clothes, and energy are being made by numbers of people around the world. And those innovations and ideas are changing the way we eat, sleep, and yes, also how we rave. In this session, you will meet some of these inspiring change makers and learn what drives them to create radical positive changes to the way we live. Now, for me, personally, this all started with a big portion of curiosity. But that wasn't always easy to deal with. As a young child, I often couldn't sleep, wondering what kind of world exists beyond the universe. Why are we here, and what am I supposed to do? These were such big questions that my little child's brain couldn't handle them. I didn't know what to do, and let's be honest, when you're 9 years old, it's also very difficult to talk about those questions with other kids. Later in high school, I found out that I was not the only one with these crazy questions, as I read about the work of Stephen Hawking and the famous Albert Einstein. Those wise men came up with theories that made me realize even more how strange but interesting the world is we live in. For example, did you know that a second on Earth is actually not the same as a second in space? And that time is relative. It depends how fast you move and how much gravity you experience. Therefore, astronauts in the International Space Station age a little bit slower than us here on Earth. I find this absolutely fascinating, but in the end, I did not decide to become an astronomer or quantum physicist, because at 17 years old, I discovered a universe I found even more intriguing. One that isn't billions of light years away from us, but that's intimately close and infinitely small. It's everywhere around us and even inside us. I'm talking about what's going on in cells, the smallest building blocks of life. And here you see a clump of heart cells, and apart from the beating, it may look like not much is going on in there, but if we zoom in, we can find an entire invisible world that is vastly complex and very lively. Deep inside the center of the cell, you can find its genetic information in the form of DNA, a type of biological software in which there are many genes telling the cell how to make thousands of different proteins. Those proteins can do all sorts of things. They give structure, transport nutrients, and catalyze all sorts of chemical reactions. Now, if we zoom in even more, we can see such a protein, and this one is called ATP synthase. It's in all of our cells, a type of biological nanomotor 10,000 times smaller than a hair that works 24/7 in our cells to turn food into energy. So when you're dancing, think about the hard work that this guy is doing for you. Another cool protein is this glow-in-the-dark one. Yes. That gives some organisms the ability to emit light. That protein turns these jellyfish into swimming disco lights. Now, when I look at this jellyfish, I just can't help but wondering how this light show is encoded in their DNA. I got so obsessed trying to understand how life works at the very small level that today I spent probably half of my life studying DNA, proteins, and cells of all sorts of organisms. Earlier in my studies, I got introduced to a relatively new field that we call synthetic biology. This field does not only try to understand how nature works, but it also tries to give cells new superpowers. 12 years ago, I used synthetic biology for the first time myself when I joined an international student competition with a team from the TU Delft. This competition, organized by the MIT, challenged students to engineer cells, give them new abilities so they could do something useful for us. And to do that, I learned about technologies to read and write DNA. With those technologies, you can transplant DNA code from one organism and transfer it to another, so we are no longer limited to only use proteins in the place where they are found in nature. For example, since we know the DNA code of this green fluorescent protein, we can send this code to a company that will print the DNA in a physical form as a type of powder, and then ships it back in a little tube to the lab. In the lab, we can use this DNA powder and deliver it to another cell. For example, to these human cancer cells that are able to read the printed DNA and produce the same fluorescent protein as the jellyfish. Now that the cells are fluorescent, we can distinguish them from normal cells and study their behavior. This technique is incredibly important for cancer research. But for the student competition, we decided to use synthetic biology to solve an environmental problem. At that time, it was 2010, there was this horrible oil spill in the Gulf of Mexico. And we saw these terrible images of animals all covered in oil. Now, even if you would remove most of the oil, what's still left are small oil molecules in water that are very difficult to get rid of. Those end up in the stomachs of plankton and other animals, including ourselves, when we eat fish. We students, we looked at this situation and we were wondering, what can we do about that? Now, with our new biology skills, we kind of felt like superheroes, and we decided to take some simple poop bacteria and reprogram their DNA to produce proteins that can break down these small oil molecules in water. After a few months of very hard work in the lab, we had a working bacterial prototype that we presented in Boston in front of hundreds of other students. Here you can see us in the middle. Funny enough, I didn't know that my future husband was there too. But that aside, and although we couldn't really, we had only something that worked in a petri dish, not a ready-to-use solution that could clean up entire oil spills, I felt for the first time in my life that I was in a driver's seat of evolution, and that I had gained skills that I could use to come up with solutions that were far bigger than myself. Over the years, I've seen hundreds of concepts and prototypes based on synthetic biology by students and scientists, and that made me see a different future. More than 10 years ago, those prototypes were seen as immature, science fiction, stuff that works in the lab, but that will never really work in the real world. But this has officially changed. The science has advanced, and those students and scientists grew up to build labs and companies that are now changing the world. Cleaning oil spills with synthetic biology is still quite difficult, but bacteria with super efficient plastic-degrading proteins have been engineered that can get rid of plastic in a matter of days. Synthetic biology companies are also producing molecules for our everyday life. One of these molecules is squalene, a moisturizer that probably most of you have put on your face today. This moisturizer comes from shark liver. And millions of sharks are killed to put this moisturizer in almost all of our cosmetics. Now, if we use synthetic biology, we can use a yeast cell to produce the same moisturizer, and we can leave the shark alone. Yes, the same yeast that you guys in Belgium here use to make your delicious Belgian beer. We can grow the yeast in low temperatures in a big tank. We give them some sugar and maybe even waste, and they will turn it into this valuable component so that we don't need to deplete the natural resources. These are just a few examples I wanted to show you so that you get an idea of what is possible. To get my doctorate degree, I used synthetic biology for the discovery of new antibiotics against superbugs, bacteria that cause deadly infections that cannot be cured. But after 4 years, I realized that I was treating the symptoms but not the cause, and that I instead should maybe use my skills to innovate the way we produce our food, and especially meat. You see, the problem is that globally, 80% of all antibiotics are actually not given to people, but to cows, chickens, and pigs. Not even to cure them when they have an infection, but to make them grow more quickly or as a cheap way to keep them healthy. Now, this overuse of antibiotics gives rise to those superbugs that can spread from animals to humans. So even developing new drugs won't fix the problem if we don't change this. Beyond the use of too many antibiotics, industrial meat production is also responsible for taking up 80% of all our agricultural land, 20% of all our water, and 14% of greenhouse gas emissions. Now, obviously, the best thing we could do is to all stop eating meat and become vegan or vegetarian. But we know that this is unlikely to happen. The world is growing, and the global world population will demand more meat, 40% more by 2050. This is so insanely, this is so much meat. Even the current meat industry cannot produce that. But if we combine synthetic biology with food science and cell biology, we can create a new type of farming that was actually predicted a long time ago by no one other than Winston Churchill. Already in 1931, so before World War II, he said that it was ridiculous to grow an entire chicken if we would only eat the wing or the breast, and that instead we should consider to grow these parts separately in a medium. Now, what Churchill predicted here now has a name. It's called cellular agriculture, and it's the use of cells to make products that usually come from an animal. 82 years after Churchill's predictions, the first cultivated burger was fried up, made by Dutch scientist Mark Post. And since then, it has inspired dozens of entrepreneurs around the world to also build cellular agriculture meat companies, cellular agriculture companies, and some of them are producing milk, bacon, or leather. And the one I joined in Paris aims to sustainably and ethically produce foie gras, a delicious product, I have to admit, but that is produced in a horrible way, whereby young ducks and geese are force-fed until their liver swells up 8 times their normal size. If we use this type of new farming, we can just take some cells from an egg, provide them with the same nutrients that they would normally find in the body of an animal. And if we farm them in these large tanks until we have enough for harvest, we can turn those cells to make this delicious foie gras that is as good as the original, but whereby cruelty is completely removed from the process. If we produce meat that way, we could reduce up to 92% of gas emission, up to 95% of land use, and almost 80% of water use. And we don't need to use antibiotics. I believe cell-based meat can therefore be an important solution to meet the high meat demand, in addition to sustainable farming, plant-based meat, and maybe even insect-based meat. But to get our foie gras into the supermarket, I'm no longer working only in the lab. Today, I work with a multidisciplinary team of food and biology scientists, together with regulatory, business, and quality experts on some very important challenges to make this happen. One of them is to mass produce this type of meat at an affordable price. And a lot of innovation is necessary, but we're making very steady progress to make that happen in the next years. Second, not every country has approved cultivated meats in their market. Singapore is currently the only place in the world where you can buy these cultivated nuggets from one company. Food agencies around the world are currently trying to figure out what is necessary to get cultivated meat approved in their region. But the big question is, of course, and I'm asking this myself as well, will people eat it? We know many people are open for it, but others hesitate, and this is completely normal. But I hope at least that you're willing to give it a try, because not changing anything is not a good option, if you ask me. That's why I want to build a future where technologies based on biology can produce what we need, but in a regenerative way, so that my 1-year-old daughter can grow up in a world where one planet is enough.